Hello, students. Welcome back. It is lab day in our topic of living things. So let us get to it right away. Yesterday, if you recall, we discussed at the end of the period 6 basic needs of living things: energy, food, water, oxygen, living space, and temperature. That's going to come in handy today as we begin our lab activity. Oops, a little preview there. Our lab activity, which has to do with testing for life. How can we test for life? How can we determine whether a sample of something has living organisms in it or not? Now, it's easy if it's a macroorganism like us that we can see and it's moving around, it's talking, it's performing all of those life functions and so forth. But what if it's a microorganism? How could we test something for microorganisms? They're living things too. Well, let's think about it. What if we provided or removed those basic needs for living things? What if we did that? Think about that for a moment and think about the lab activity that we're going to be performing today on page 34 of your books. The problem is, how can a soil sample, how can a sample of soil, how can soil be tested for microscopic living organisms? Now you can look outside and see whether there's a groundhog coming out of the soil, or a mouse, or a mole, or a deer roaming across its surface. But how could you test a sample of soil for microscopic organisms that might not be that easy to see? Think about that and write a hypothesis. All right. Well, I kind of gave you an idea here with that previous question. What if we provided or removed the basic needs for living things? Could we observe changes taking place? Well, what if we wanted to test the soil from another planet for living things? What? What is Mr. Skirps talking about? Well, today's lab activity, you may have anticipated we were just going to test some earth soil for living things. But as I mentioned to you earlier in the week, I have connections at NASA. So, NASA has charged us here, students at our school, with a very important mission to help them out. So let me give you a little backstory here. We mentioned earlier in the week about the red planet Mars and how there may be water on Mars, and there certainly is plenty of evidence to suggest that there was water on Mars long ago. And there has been a particular location on Mars of real interest to NASA scientists: Gustav Crater. We've got a mission headed there right now. And we've sent other missions to the surface of Mars also. And we've sent rovers that look like this one. We have one on its way. We have a few of them there already, and they are charged. And here's an actual picture from the surface of Mars. You can see this is a solar panel on a rover, and here is a claw to remove soil samples from the surface of the red planet. This is an actual photograph. And here is the collection site on the planet Mars where our samples have actually come from. Now, if you want to see something cool, I'll show you a little video of how the spacecraft got to Mars in the first place. Check it out. This was the Opportunity mission to Mars quite a while ago, and it traveled through space tens of millions of miles through space to the planet Mars. Now, even though Mars's atmosphere is much thinner than Earth's atmosphere, it does have an atmosphere. So as the spacecraft was traveling through the atmosphere, it needed a heat shield to protect it from the immense heat of entering the atmosphere. And then the parachute deployed. And yes, it needed to be a bigger parachute than here on Earth because the air is so thin, but a parachute nonetheless. The heat shield was released, exposing the spacecraft itself to a Martian landing. But get a load of how Opportunity landed on Mars. Whoa! It blows up like a bunch of grapes. No, they aren't grapes. They are airbags, very strong airbags. And watch how it strikes the surface of Mars like a gigantic beach ball. Bouncing, bouncing, bouncing. This is how the Opportunity landed on Mars. Gravity pulled it until it eventually rested upon the Martian surface. When it was positioned just right, the balloons deflated and it deployed the lander. The Opportunity. The solar panels deployed, allowing the batteries to charge, and voila, it took some self-portraits to show us that it survived the landing on the red planet, and then it went out to seek for soil samples. Check it out. Is that cool or what? And this is all done remotely from tens of millions of miles away as the Opportunity spacecraft travels around the Martian surface looking for the most ideal location. Listen, may have heard the wind. Listen. Yes, the Opportunity has microphones. You can hear the wind on Mars because, again, there is an atmosphere, albeit thinner than Earth's. But the spacecraft then went around the surface looking for samples. And again, this is what the location of the sample spot appeared. And that brings us to today. We have those samples in the lab right now. So I am going to move over to the laboratory for a close-up view, and we are going to investigate for you what is located in this box. So first, let me open the box carefully. It has just arrived from NASA, as you can see from the labels. I'm going to remove the contents here. And inside this container are a set of goggles we have to wear first, because safety is the number one priority. And we want to protect ourselves from any alien creatures. So I am going to be wearing gloves here. And as we perform the steps of this lab, you are going to be required to record your observations to come to a conclusion. So there's information here as to the location from Mars. Here's a map of Mars and exactly where the sample was collected from on the Martian surface. And here we go. We have 3 different sampling locations. Now they're just being identified for us as A, B, and C. And I believe you can see these. I'll put them on a different colored bottom here. All right, I think we can see these now. So there's sampling locations A, B, and C, and NASA has asked us to test these 3 soil samples for any evidence of microorganisms, living things from Mars. Now, in order to do this, we're going to modify our lab procedures just a tad, and we're going to apply those procedures to our experiment. Now, think about the problem again. How can we test the soil sample, even though it's from a different planet? How can we test it for living things? Maybe it has to do with providing some of the basic needs of living things. So what were some of those needs for living things? You can pause me for a moment as you discuss as a class those needs for living things. Okay, we're back here, and I have some materials in the lab that we're going to use to test these samples. I have water. Water is a basic need of living things, and I've also heated up this water to an appropriate temperature that, at least we're aware of on Earth, most microorganisms prefer. Body temperature. This water is body temperature. So we've got water at an appropriate temperature. What about food? Well, as far as we know, all life on Earth desire the simplest form of food, sucrose, or glucose, or fructose, or lactose. All of those words that end in OSE are types of sugars. So I have some sucrose here, which is a general type of food source. NASA gave us a special supply. I just wanted to show you what sucrose is. It's white powdery substance, but they gave us glucose. We're actually going to use what NASA provided for us and labeled it food, and it is nothing more than a type of sugar. And I'm going to add this glucose into our water, which is at an appropriate temperature. And they gave me specifications of how much to add, so I'm following NASA's instructions as well as the instructions in our lab. And I'm going to agitate or stir this sucrose, or this glucose rather, the sugar, into the warm water. So we now have 3 of the basic needs of living things in this beaker. We've got water, warmth, and food. But there are a couple of other things that we need to provide some basic needs. So I'm going to put this beaker off to the side for a moment, and I am going to bring over a test tube rack. A test tube rack with 3 test tubes labeled A, B, and C. I am providing here in these test tubes a living space. And into each of these living spaces, I am now going to place some of our soil samples. So here is soil sample A. Okay, and I just have a paper towel here. I'm just disinfecting, decontaminating the scoopla each time. Let me put sample A away for now. Soil sample B in test tube B. So we've got our second soil sample in a living space potentially, assuming there are any living things in it, and soil sample C that I will place into, this one has an odor to it. Soil sample C into test tube C. Okay. So now I will arrange these so that hopefully you can see them. Sample A, sample B, sample C. Now right now they only have a living space, but what we want to add to them are some of the basic needs of living things. So again, in here, we have water, warmth, and food. We're going to add these things into the test tube, and then I'm going to put my thumb on top of the test tube and shake it. By shaking each test tube, we're going to be adding the last basic need of living thing, some oxygen. We're going to be introducing some oxygen into that living space with the other needs of living things. Before I do that, however, I want you to think for a moment. If there are chemicals in here, if there are living things in here, if there's nothing in here other than mineral soil, I want you to consider what observations we should be looking for. You can pause me for a moment and look at your lab procedures. There is a note in there for you to read before I add these basic needs to our test tubes. So pause me now and discuss. Okay, I'm back. So here we go. I'm going to add now our warmth, our water, and our food into the living space of each of these soil samples. And then I'm going to agitate and add some oxygen, and we'll make some observations. So we've got all three of them ready. Here we go. Test tube A, test tube B, and test tube C. Now, I'm going to use my senses on your behalf, since it's difficult to use all of your senses on camera. And I am now examining, we'll start with test tube A. Test tube A, hopefully you can see this, you can make some observations yourself. All of the soil sample from test tube A have settled to the bottom. They're an interesting color, kind of an orangish, purplish color. Looks like sand. But nothing else is happening. The water is just doing nothing. A little bit cloudy, but mostly clear. So there's not much of anything other than settling sand in test tube A. Test tube B. By the way, test tube A, no odor whatsoever. Test tube B. There is a lot of settling on the bottom, but initially you can see how cloudy the water is. That's because there were a lot of bubbles that were released very quickly after I put the water in and shook it. There were a number of bubbles that came up, but now the bubbles have stopped. No more bubbles. So bubbles initially, but no more bubbles. And really no odor. No odor detectable by me. And now test tube C. Test tube C. There was some settling of solids on the bottom. The water is very cloudy, and it's also a little bit foamy on the top because it's still making bubbles. There are bubbles actively being produced right now. There's some foam on the top with active bubbles. And, whoa. Okay, there is definitely an odor for test tube C. No doubt about it, as it continues to bubble. So to recap, test tube A observations, settling of sediment, no bubbles, kind of boring. Test tube B, bubbles initially, but no more bubbles now, and just settling, no odor. Test tube C, it's still making bubbles, and it stinks. Oh, I wish you could smell it yourself. I really do. Woo-wee. Yes, I do wish you could smell that. But it does have a very pungent, distinct odor that I think I have smelled before, or at least something like it. So let's think about this as we go back to our lab activity. Let's go back to our lesson notes here, shall we? Let's talk about our observations. Testing for life, whether they are in a soil from Earth or a soil from another planet. What did we provide our samples? What were some of the things we provided? Basically, the basic needs, right? We provided a living space, water, warmth, food, oxygen. In other words, I just wanted to go back a slide here. In other words, we were providing these basic needs of living things. The only one that we didn't add, essentially, was energy. But we are surrounded by energy right now. So after adding some basic needs of living things, using that note in your lab book, what could we conclude? Take a moment to think about your observations. Read that note there after step 6, and let's take a few minutes to discuss your findings. So you can pause me and discuss your findings. Okay, I'm back, and we have discussed your findings. So by a show of hands, how many of you thought there were living things in test tube A? How many of you thought there were living things in test tube B? And finally, how many of you thought there were living things in test tube C? Well, if you said you believed that there are living things in test tube C, you are absolutely correct. That is what this test has determined. There are, in fact, microorganisms in test tube C. There are. They're living things in there. All right, now before you get too excited, there is some truth. Well, there is some fiction and nonfiction in today's lab activity. First, the nonfiction. Yes, I do have an uncle, although he passed away a number of years ago, that did work on the lenses at NASA for the Viking spacecraft. And his name is on the Viking spacecraft, which is pretty cool. There are 2 of them, Viking 1 and Viking 2, that have his name on them. That's pretty neat, I think. Another truth of this story is that the Viking spacecraft, long before the more modern spacecraft in the 90s and the 2000s landed on Mars, performed this very same test that we just did in the lab. This is a test that was performed on Mars back in the 1970s. And guess what they determined? They didn't find any living things. They added the basic needs for living things, and they didn't find any living things. Now, what's the fictional part of this story? Well, to date, as of the time of this story, no samples from Mars have ever been retrieved and brought back to Earth. I'm sad to say those samples that we tested in the lab were simply playground sand from my backyard. Test tube 1, test tube A, was just sand. Test tube B had sand and some baking powder in it, some sodium bicarbonate. That's what caused the little chemical reaction initially, and then nothing resulted. Test tube C, this is true, there are living things in it, but the living things are unicellular organisms that are found here on Earth called yeast. There were yeast cells in test tube C. So we did perform an experiment testing for living things, and our results were correct. There are living things in test tube C. It's just that these samples are not from planet Mars. Now, before you become too disappointed, however, NASA is currently, and this is true, is planning several missions to Mars in the very near future. Actually, they're planning this in conjunction with the ESA, the European Space Agency, as well. And these different agencies are sending another rover to the Martian surface, which is going to go around and collect samples. Later on, another mission is going to send another rover to Mars that's going to pick up these samples, which will be like little test tubes, and actually take off from the Martian surface with them and bring them back to planet Earth. So keep an eye and ear out in the news, because these missions will be coming up in the relatively near future. So pay attention to Mars news, and perhaps someday you might actually be performing this experiment here on Earth for real. Who knows? So stay tuned. So I hope you are able to complete your observations and write a conclusion before you pack it in for the day. But I'm going to give you the rest of your time to finish up what you need to finish up in your book, as I remind you one last time about tomorrow's quiz. Be sure to study. Hit that help button on Google Classroom to help you study for tomorrow's quiz. And I'll say bye-bye for now.